Hello everyone, welcome to Enzyme MBBS, where complex medicine becomes simple and exam ready. So in this video, we are going to discuss necrosis as a long essay question, exactly the way it is expected in MBBS examination. Because as I shared in the last video, the only long essay question that comes from this chapter is necrosis. So looking into the question is like, define necrosis, mention types, explain causes and pathology of each type of necrosis with example, and difference between necrosis and apoptosis. So we are exactly going to write the same answer. So coming to the answer, first of all, definition. Now the definition is, which I have given here, is darkly highlighted in the Robbins. Now as soon as you write this definition in the answer, the examiner will come to know that okay, this guy or this girl is reading the Robbins and she has written this. Now imagine the first question in your paper, obviously it will be a long essay, and if you write the direct definition from the Robbins, the examiners becomes mentally prepared to give marks. So that is how you make the examiner in your side even before the answer paper is getting to start corrected. So coming to the definition, it states necrosis is a pathologic process characterized by irreversible cell injury leading to cell death with denaturation of cellular proteins, leakage of cellular contents through damaged membranes, local inflammation and enzymatic digestion of lethally injured cell. Now some of you may find this little complicated, but it is very simple. You need not memorize it. It is just like, see, it is a pathologic process, means something is wrong. It is never physiological, it is always pathologic. What is its characters? It is basically happens when an irreversible cell injury takes place. Now obviously injury is bad, and if it is reversible, then it basically means that we can correct that bad. But this necrosis basically happens when something bad happens which cannot be corrected. So that is basically irreversible cell injury. So that will lead to the cell death. What happens there? When the injury takes place, the cellular proteins get denatured, because of which the contents inside that cells gets leaked out through the damaged membranes. Now not only the cell membrane will get damaged, inside various cell organelles are present and their membrane also gets damaged and their inside contents also gets leaked. So there is something organelle called as lysosomes, which all you know, which are basically suicide bag of cells. Now even their enzymes which is present inside it, that also gets leaked, and all the other contents also gets leaked, right? So that obviously calls the WBC and all other things to accumulate in that particular place, which leads to the inflammation. That's why they basically say the next event that happens after necrosis is inflammation. So that local inflammation takes place, and then the enzymatic digestion, which have been the enzymes which have been leaked out, they basically digest that lethally injured cell. So that's all that is given in the definition. So there we complete the definition. Then coming to the general causes. See, first, the most common cause is ischemia and hypoxia. What basically happens in ischemia and hypoxia? You all know that in the hypoxia or ischemia, basically oxygen level goes down. Now, we have studied something called as electron transport chain in the biochemistry in the first year, which basically very important in the production of ATP. And it also requires oxygen in the electron transport chain. Now when the oxygen level goes down, this ETC function gets hampered and ATP production comes down. Now we have also studied something called as sodium potassium ATPase pump, which basically involved in the ionic exchange. Now that requires ATP, right? Now when ATP level goes down, normally the sodium is more outside and potassium is more inside, right? Because of this pump. But since ATP level is going down and this particular pump function is hampered, the intracellular sodium level goes above. Now we all know that close friend of sodium, that is water. Wherever the sodium goes, water follows. Now the same happens here also. The water comes inside, and as a result cell begins to swell. It swells and swells, and at a point it will burst. So that is what basically happens. That is why we said that ischemia or hypoxia is the most common cause for necrosis. And the second cause is physical agents. Now under physical agents you can say like trauma or heat or radiation, all these things causes the necrosis. Next, chemical agents and drugs and exposure to some microbial toxins, all these results in the necrosis. Next, general morphology. Now, this general morphology, they haven't asked this particular thing in the question, but you can mention in the answer because it involves a diagram, and as soon as you put a diagram, okay, the examiner becomes happy. So in order to include that diagram, which I have shared in the video, you can just write that, and it will also give you that extra marks. So coming to the under general morphology, we will write two headings, that is, one is cytoplasmic changes and one more is nuclear changes. Now, cytoplasmic changes, you can see it in two ways. You can see it in the light microscope, you can see it in the electron microscope, and I have mentioned both things here. So coming to the light microscope, which have been the drawing I have been drawn, it is from Ramdas Nayak. So there you just draw that and show that increased eosinophilia and moth-eaten appearance, which you can see which are the findings under light microscope. And also see something called as calcification we will be studying in this chapter. Most of you will be aware of it. So this calcification happens in all the necrosis after a point of time. So here also there will be dystrophic calcification that is seen in the later stages that you can find, and you can also see something called as myelin figures. Now we all know that in the cell membrane there is a component called as sphingomyelin, right? This myelin figure is something associated to that only because cell membrane gets damaged, right? So that is one thing. And third thing is amorphous densities in mitochondria. Now the name itself states amorphous, means the mitochondrial morphology is affected and it has become amorphous because of necrosis. That is one more thing. And pycnotic nucleus, that is nucleus size has been shrunken. So this is the cytoplasmic changes. And coming to the nuclear changes, the nucleus obviously first pycnosis, then karyorexis, then karyolysis. Basically, first nuclear gets shrinked, then the fragmentation of nucleus takes place, and ultimately it gets dissoluted and it disappears. So that is pycnosis, karyorexis, and karyolysis. So this is the general morphology of a cell that is that have been undergoing necrosis. Now coming to the types of necrosis. Before explaining each type of necrosis, just write all the 6 necrosis types. In that, obviously one necrosis is not actually a distinct histologic type, but you can mention it here. So they are coagulative necrosis, which is the most common. Then comes liquefactive, caseous, fibrinoid, fat necrosis, and gangrenous necrosis. Now gangrenous necrosis is what I mentioned. It is not a distinct histologic type. It is just a clinical term that is used. And fat necrosis can be further classified into 2 types, that is enzymatic necrosis and traumatic necrosis. Now let's explain each particular necrosis. So coming to the coagulative necrosis, before that we will write all the 6 necrosis under few common headings, that is cause, pathogenesis, morphology, and examples. So first, coagulative necrosis. What is the cause for it? As we said, the most common cause is ischemia and hypoxia, and the most common necrosis is coagulative necrosis. So the same cause happens here, that is ischemia and hypoxia. This is the most common cause, and coagulative necrosis is seen in almost all solid organs except brain. In brain something different happens that we will see in the same video. Next, coming to the pathogenesis. Now pathogenesis, what basically happens? Now we saw that when the oxygen level comes low, cell bursts and everything that happens. So basically during the cell bursting, we know that the structural proteins and enzymes gets denatured. Okay, that gets denatured, and that basically blocks the proteolysis of the dead cells. What it does, it blocks the proteolysis of the dead cells. So when we see under the microscope, we can see that intensely eosinophilic, because the proteins will be seen under eosinophilic stain. So it has intense eosinophilic cells with indistinct or reddish nuclei persistent for days or weeks, because nucleus have undergone that all those pyknosis, karyorrhexis and karyolysis. Now coming to the morphology. Morphology, you have to write gross and microscopy both. Now the gross, it is basically dry, pale, firm and yellow, and infarct is well defined, that is due to ischemia. And the name coagulative comes because this appearance, the gross appearance, resembles coagulation of proteins which occurs upon heating. You would have done that experiment in the biochemistry in the first year in the urine analysis, that you heat and acetic acid test, the protein precipitates, right? So that is what actually happens. Now coming to the microscopy. Microscopy, it is like the classic finding of coagulative necrosis is we say that the tissue architecture is preserved. What does that basically means? See, in few other type of necrosis which we are going to study next, there the complete cell structure goes away. You cannot identify the cell border or something like that. But in coagulative necrosis, we can identify the tissue architecture. We can see those cells, but we can also say that it is dead. It's something like that. So we call it as tombstones. Okay, it's like in the tomb, the dead person is present, but he is dead. It's similarly like that. Next, the loss of nuclei also is seen, that is karyolysis, and eosinophilic anucleate cells, because nucleus undergoes the karyolysis, right? The examples for this are, as we said before, all the solid organs undergo coagulative necrosis. So the myocardial infarction, renal infarction, splenic infarction, all are the examples of coagulative necrosis. Now coming to the liquefactive necrosis, here what actually happens is, this is mainly due to bacterial and fungal infections. What they do, they basically digest these dead cells. So they are transformed from tissue into viscous fluid. So that is why the name liquefactive. So this is the most common cause, and in coagulative necrosis we discussed that brain does not comes under this type. Its necrosis happens in a different way, and it happens in this way, that is liquefactive necrosis. So hypoxic injury to CNS. Now coming to the pathogenesis, as we discussed before, these microbes, what they do, they stimulate the accumulation of leukocytes and liberation of enzymes from the leukocytes. Now that enzymes will digest these dead cells, so there will be a liquid mass. So coming to the morphology, gross, it is basically soft liquefied tissue, and pus formation takes place because of the microbial infection. And microscopy, here you can say that tissue architecture is lost. In coagulative necrosis, tissue architecture was intact, but here it is lost. And of course, you can see numerous neutrophils here and the granular debris. So the examples for this is brain infarction and abscess. Abscess is nothing but localized collection of pus. Coming to the caseous necrosis, the cause for this is basically, this is basically, even they ask in Viva, that is, what is a classic finding, where you can find the caseous necrosis? It is tuberculosis. The most common and obvious answer for this is tuberculosis. It happens due to type 4 hypersensitivity reaction, which we are going to study in immunology. Now coming, some other causes is some fungal infections. Coming to the pathogenesis, this is basically a combination of coagulative and liquefactive necrosis, and it is associated with something called as granulomatous inflammation. Now again, this is, we are going to study this granulomatous inflammation in the upcoming videos. For time being, just remember it. Now coming to the morphology, there is gross and microscopy again. So gross, it is soft, friable and cheese-like. That cheese-like word has very significance here. The name caseous comes from the same thing, cheese-like. And microscopy, it is amorphous granular eosinophilic debris, no cellular outlines, and surrounded by epithelioid cells and giant cells. Now these epithelioid cells, giant cells, all this is something related to granulomatous inflammation. Now it's a separate big topic, so we will limit us here for only this thing. Now coming to the examples, it has tuberculous lymph node and pulmonary tuberculosis. Now coming to the fourth type, that is fibrinoid necrosis. Now anything oid means like. So fibrinoid means fibrin-like. Now where do you find fibrin? It is something related to vascular structures, right? Blood vessels. So the causes is obviously the first common cause for this is immune mediated vascular injury. Okay, there is something called as polyarteritis nodosa, which again we are going to study in systemic pathology. So that is something where you can see fibrinoid necrosis, and other cases are malignant hypertension. Now coming to the pathogenesis, what actually happens here is, see, something immune complexes will get deposited in the vessel wall. Immune complexes means antigen, antibodies, something like that. It gets deposited in the vessel wall. Now this deposition, along with the plasma proteins which have been leaked out from the vessels during the cell death, they both result in a bright pink amorphous appearance in H&E stains, which looks like fibrin. So they give the name fibrinoid necrosis. Now coming to the morphology, here you can't see gross because it is inside the blood vessels. You can't observe it as a gross. So here we only mention microscopy. So obviously you will write looks like fibrin, hence fibrinoid, and bright pink amorphous materials. Examples have listed few. 2 or 3 will be enough in the exam. So the examples are polyarteritis nodosa, malignant hypertension, and there is something called as ACE of bodies in a particular disease called as rheumatic heart disease, which we are going to study in the cardiovascular pathology. So that is one thing, and placenta in preeclampsia and hyperacute transplant rejection. So all these examples comes under fibrinoid necrosis. Coming to the fat necrosis, as I said, the fat necrosis can be classified into 2 more types, that is enzymatic necrosis and traumatic necrosis. So traumatic necrosis means there is something trauma to the adipose tissue. Adipose tissue is the tissue where the fat gets stored, right? So trauma to adipose tissue, that is one cause. And one more is release of fat destruction enzymes. That is what enzymatic necrosis, which you basically see in pancreatitis. Now pathogenesis, see, in the SNR cells, the pancreatic enzymes are present. Now, something damage happens to the pancreas, and that SNR cells gets damaged, so that the pancreatic enzymes gets released. Now, when it gets released, it results in the hydrolysis of triglycerides. Now, triglycerides hydrolysis, after it becomes free fatty acids, right? Now, some minutes ago, I mentioned that all necrosis will undergo calcification after a point of time. That means calcium gets deposited. Now the free fatty acids are present, calciums are present. Now they both combine and form some salts. Now this is known as fat saponification. Even basically it's a kind of soap. Okay, so that happens because of hydrolysis of triglycerides into fatty acids, which combine with the calcium, and that results in fat saponification. Coming to the morphology, gross it is chalk whitey areas, and microscopy it has shadowy outlines of fat cells, basophilic calcium deposits, inflammatory reaction. Examples are acute pancreatitis, it is basically example for enzymatic necrosis, and traumatic necrosis example is traumatic fat necrosis of breast. Now coming to the gangrenous necrosis, see, the gangrene itself is a big topic. If we want, we can discuss it for an hour. But here, it is just a small, it just needs a small mention in the 10-mark question. So I've given a gist of it. We will write the answer for the gangrene separately as a 5-mark answer. So here it is enough. And if you want to write little more, watch that video, there the answer will be present. So you can add whatever you want. So coming to the gangrenous necrosis, it is a massive necrosis with super added putrefaction. Types, it is, see, we can classify it into dry gangrene, wet gangrene, and gas gangrene. But moreover, all these gangrenes are a variant types of the necrosis which have been already discussed. For example, dry gangrene, it comes under coagulative necrosis itself. The example is ischemic lower limb. Again, the cause is ischemia, which we discussed in the coagulative necrosis. Similarly, wet gangrene. Now, the example is wet gangrene of small intestine. There again, basically that pus formation, all things takes place. So, it comes under liquefactive necrosis. And gas gangrene, it is basically a variant of wet gangrene, which is caused by a gram-positive bacteria called as Clostridia. So this much is enough about gangrene here, because it is, as we discussed before, it is just a clinical term that we use. It is not a distinct another histopathological type. So that is enough. And coming to the differences between necrosis and apoptosis, again, when you write a 10-mark answer, you have to cover all those things. So you will write everything a little for it. But the differences between necrosis and apoptosis is a separate 5-marker question. Here I have given only the tabular column. There we have to explain this tabular column, and we will write the answers for the same thing. We will do the same thing in upcoming videos. So in this answer we will limit ourselves for this tabular column only. And one more thing is, to understand the apoptosis column in this, we have to study apoptosis. So when we discuss the apoptosis answer, I will come back to this table and I will explain it once again. Now we will just see the necrosis table and the features. So while writing the difference, you have to write the causes, biochemical events, lysosomes, cell size, integrity of cell membrane, nucleus, and adjacent inflammatory response, DNA electrophoresis, and tunnel staining. This table is basically also given in the Ramdas Naik. So necrosis, see, what is the cause? It is invariably pathological, right? As we discussed, necrosis always happens, then there is a pathology. But apoptosis is not like that. It is a physiological programmed cell death. Sometimes apoptosis may also be pathological, but more often it is a physiological process. Biochemical events, it has written that impairment of ion homeostasis. Why it has written? We discussed this, that ETC, electron transport chain, gets hampered because of the less oxygen. So sodium, potassium, that ion thing gets impaired, and all those ion problem happens. So that is why it has written impairment of ion homeostasis. Whereas apoptosis, it is something different. And lysosomes, in apoptosis it remains intact, whereas in necrosis it leaks the lytic enzymes, and because of that digestion happens, right? And cell size, in necrosis it is basically swelling happens, cell becomes enlarged and then it undergoes lysis. But whereas in apoptosis it shrinks. So then integrity of cell membrane, in necrosis it is disrupted, but in apoptosis it is maintained. So when the disruption happens, so the inflammatory response happen. Sorry, there is a mistake in this table. I have written none under necrosis and usual in apoptosis in adjacent inflammatory response. It has to be reversed, because inflammatory response is present in necrosis and it is absent in apoptosis. So make that correction, okay? And then nucleus, yeah, in necrosis it undergoes pyknosis, karyorrhexis, and karyolysis. Apoptosis, fragmentation happens. And electrophoresis and tunnel staining, this I will explain in the that 5-mark answer of necrosis apoptosis, because it takes some time. Now the video is already long for 22 minutes. So yeah, for time being, you just remember it. I will explain in that video. So that sums up the complete answer of this necrosis question. So yeah, write those Robbins definition, and basically this answer has a mixture of both Robbins and Ramdas Naik, and present it in such a way that examiner becomes happy. Include the diagrams, write the main headings, write the underlines, and while writing the answer, the keywords, you can make a cloud around it or underline it. All these makes easy correction for the examiner and he give the marks. So that's how it goes. So then we end this, and I have a request with all of you guys that comment on this video. Whatever that comes in your mind, just comment, because it helps us to grow. It helps us to reach the more audience. So we need your support on that. So comment, and yeah, keep following. This is just a trailer. Only one question we have been discussed. There is plenty of questions in all the subjects. All the videos are coming in upcoming days like this, a proper answer, which will fetch you very good marks in the exam. So yeah, keep studying. All the best. Do well. Thank you.